Hello, everybody this afternoon. This is a small crowd. So you guys, as I'm talking, if you have questions, please just interrupt me. Do you hear me or do I need to? Okay. So first of all, I want to set the stage straight. I am not dead. That was not my obituary, but but it kind of sounds like it. I've been interested in anti-infectives for quite a while, and I'm pleased, or I shouldn't say pleased, but I want to tell you I'm very upset I accepted this talk because this was a difficult talk to do. I will never do one of these again because as I was presenting this, 5 drugs became approved that were in my pipeline. So that means I had to change the talk again. I called up Shannon and I said, how long do I have before I have to actually submit something to you? Because the marketplace kept changing. So I want to try to give you like a overview of what I think is going on out there. I think there's some very exciting things, but we still have areas that are very bad for us. So, and I'll go talking about this. So I'm going to talk about it in the essence of what do we know about antimicrobial resistance, what's happening, what are the therapies we might see in the future, moving away from typical anti-infectives that we're all familiar with, such as beta-lactams or inhibitors, and looking at some of the non-traditional therapies. And then I also want to spend a little time talking about the problems we're going to have as we move forward with this in the future of what is, we may develop drugs, but what's going to stop us from developing drugs. So one thing to remember, bacteria have been around a long time. So that means we like to anthropomorphize and say, oh, bacteria are really smart and they get resistance. So as soon as we developed antibiotics, almost immediately you saw some issues with resistance. And if you take a look at this, and hopefully you can download the slides because this is busy, but basically on the left is when an antibiotic was developed, and on the right when resistance appeared. So as you can see, basically resistance occurs generally within 1 to 2 years from when a drug is approved. And I always used to love when a pharmaceutical rep would come into my office and would tell me we have a new antibiotic and there's no resistance. And I said, okay, wait 6 months, come back and tell me again because it's only a matter of time. Probably about one of the few drugs that I think about that it took quite a while to develop resistance, and I really don't know why, is vancomycin. Vancomycin resistance didn't occur immediately, but it's one of the very few drugs where you see it. So the bottom line and the way it works, antimicrobials are very effective in trying to figure out how to mutate to overcome the new drugs that we develop. And there's a problem. Yes. No, you're right. It has not, but you can see some resistance, but not bad. We still have effectiveness. No, we know what? I don't know of any people who are doing research to looking at some of our molecules that we have to try to determine why this resistance is not occurring. So that would be if you're interested in research, there's a good line. Let me know, and maybe we'll work something out. So some of this data is like from about 2019, 2020. The bottom line is a lot of people die from resistance associated with it and actually exactly attributed to it. In the US, we estimate that we get 3 million resistance infections a year, and about 35,000 deaths. So it's a major player. And although we don't like to talk about finances, it's the cost of resistance to our healthcare system is in the billions of dollars. Now, the issue we have is that over time, the antibiotic pipeline is now small, and it is very difficult to get something approved, so it's actually very fragile. At this point, over the last decade, there's only been about one antibiotic a year, new antibiotic a year developed. When I was in the beginning of my practice, so talking about the 80s and 90s, we usually had about 10 different antibiotics that were being developed every year by pharma. We don't see that anymore. Pharma doesn't want to invest in an antibiotic. What does pharma want to invest in? They want to invest in the next Viagra, right? Because you can keep selling that. Antibiotics you can't sell because antibiotics are one of the few classes of drugs that as you use them, they are going to become self-limited. right? That's what resistance does. We use them, they become resistant, and then you can't use them again. So, like I said, so it was different than in the 80s and 90s. Actually, we used to, when resistance would occur, right now, when we have resistance, we have all these programs on stewardship, right? We're trying to figure out how to maintain it. Back in the 80s and 90s, when resistance was occurring, we didn't have stewardship. We just waited for the next drug to come out. So in those days, we actually called it, what's the next chef du jour, right? The next cephalosporin that's going to be developed, and we would just switch to that because it was active against these resistant pathogens. We don't have those luxuries anymore as we had in the old days. This kind of shows you. This is a compilation of some different graphs. By the way, ChatGPT helped me with this. I took three different figures and I said combine them, and it worked pretty well. So I'm declaring that AI did help me on some of this. So the bottom line is you can see drugs are declining and resistance continues to increase. That's not a good strategy. The WHO has been very active in trying to help and focus our attention on the fact that we have antimicrobial resistance issues and that we have a minimal, limited supply of drugs in the pipeline. And they're trying to encourage pharmaceutical companies and governments to really put resources into this to be able for us to combat antimicrobial resistance. And if you take a look at the total number of compounds that are in development, they have been decreasing over time. And again, as I said, the reason for that is pharma does not see antimicrobial therapy as a way to increase their profit, increase their bottom line. So they're looking at different antibiotics altogether. So if you take a look at this, the WHO has done analysis on potential pipeline drugs. This one was in 23, and the last one they did was in 25. So they had 97 agents in development, and you can see 97, 4 were approved, 10 were withdrawn, and only one of the 90 was in regulatory review. Today we have 90, so there's just a little bit fewer. But what's happening is the innovations are shifting away from our traditional antibiotics. That's the way of the future, really. It's going to be traditional antibiotic with a non-traditional approach or a non-traditional approach by itself. So if we take a look at these 90 antibiotics that the FDA, the WHO characterized, 15 are only considered innovative. So that means the other 85 are really basically traditional antimicrobial type compounds, which means we know that they're going to be limited relatively quickly. And of these 90, only 10 have available data that looks at the potential for cross resistance. So that means that a drug, one of these new compounds could be coming out, and they could immediately develop some cross resistance that we didn't think about. And of these compounds, so of these 90, only one is addressing what the WHO calls critical bacteria. And in a few seconds, I'm going to show you a table that looks at it. But the WHO and the CDC have identified microorganisms for us, bacteria for us, that we should be really concerned about. This shows you where we are today in terms of the phases of research for different drugs. So you can see a lot of drugs in preclinical development, and everybody's probably, pharmacists at least in the room, are aware that of all these compounds, a very few number will ever make it into a clinical trial. And you can see here, all we have really, a few in phase 1, which is looking basically at safety. Phase 2 is looking a little bit more, and only 18 out of these compounds have made it into a phase 3 trial. And it's the phase 3 trial data that we use that ultimately will tell us if a antibiotic is going to be approved or not be approved. This is the table I was telling you about. So these are the pathogens that the WHO and the CDC say are critical organisms for us that we have to develop therapies for. What I didn't put on the list, I left off gonorrhea. We know that, as everybody is aware, gonorrhea, very big problem internationally because of the high rates of resistance. Some new drugs have just come out, so that's pretty good. But the WHO, and in their 2025 report, are saying pharmaceutical companies and researchers should be focusing themselves on these agents, right? I mean, these bacteria, looking for crabs, CRE, ESBLs, and so on, because these are the bacteria that we don't really have good drug therapies for. And as I said, so what we're looking for is, are there new antibiotics we might be able to use? Or what basically re redeveloping or adding a new chemical moiety to one of our other compounds, or what are some non-traditional therapies? So this is just a table to give you an idea of drugs that are out there that do meet the need of the WHO for their urgent compounds. And just to highlight what I'm saying is the first agent, solithromycin, which was being developed, is already not in development anymore. It's a ketolide. Ketolides have a lot of toxicities that we already knew about. The company tried to develop it, Nabriva, and when it got to the FDA said, no, we're worried about hepatic toxicity. We're not going to give you approval. But these drugs, and I'm going to talk about these drugs a little bit more specifically as we go on. So what we're worried about is that we're seeing a shrinking pipeline, and we're seeing more resistance. And there's there's economic challenges to that because nobody wants to invest in it. Because why is that? There are problems what we need. So the idea is maybe we need to have new models and mechanisms for the economics and developing antibiotics. And part of this, what the WHO has put out there, and actually they're looking at this in England, is that where the government is going to subsidize companies to develop antibiotics. So a company will know they will be guaranteed a certain amount of money for a certain period of time if they develop an antibiotic. Because what happens, develop an antibiotic. I assume everybody here is on a stewardship team, right? And what's our job? Our job is to tell everybody in the hospital, don't use the antibiotics, right? So you run into that brick wall. So the company is looking at it and go, well, why am I developing a new drug if everybody's going to tell me I shouldn't use it? And actually, what do we do when a new antibiotic comes out? Most of us do what? We restrict it right away. All right. At our place, when it first come out, so we're at UIC, we're like 2 blocks away from Rush. So what we used to do is UIC, we would say we're not buying it. And they say, well, what if somebody gets sick? We go, well, Rush puts everything on formulary. So we'll just ask them to send it over to us because they're 2 blocks away from us. So Rush was basically our non-formulary drug depository. But we need different models. But the issue is really, if you look at it, market failures, it really hits you in the face of how difficult this marketplace is. So here, as I said, here are some of the drugs that were recently approved. And these were the drugs I had already written slides on and how they were in the pipeline. But, as fast as you do something like this, already the sulopenem is already not available. What happened? Sulopenem is made by a company called Idph Pharmaceutics. And Idph Pharmaceutics got their approval, but they're a small company. And when the drug came out, people were limiting it. So they were not making back their investment. So basically, they no longer exist. And here, just from about 2019, these are pharmaceutical companies that developed a new antibiotic and are no longer in the marketplace selling these antibiotics. And because the model has shifted from when I first started, where it was the big companies, Pfizer, Merck, Upjohn, you probably don't remember them, Upjohn, other pharmaceutical companies, the big ones, they were all developing antimicrobial agents. The reason they were developing, because we were misusing antibiotics. It's unbelievable when you can think about it. When azithromycin came out, it made $1 billion a year. Clarithromycin, $1 billion a year. some of the earlier quinolones. And that's because as soon as they came out, we were using them no matter what. We used them like crazy because, like I said, if something became resistant, we went to the next one and the next one. So we used them. But now, as you know, everything has kind of shrunk down. And so Big Pharma has decided they're not interested really to do this. So what they do is they let a small company develop it. The small company gets investors. If the product hits, they may buy it. But in many instances, like in these, nobody picked up these compounds, and so we've got these new drugs that are not available to us because the companies went out of business before they can really establish a foothold to say how effective it is. And some of these are very good compounds, and so to me, it makes sense if we do have an arrangement with the government or some kind of entity where a company is guaranteed if they develop a new product for one of these MDROs that we have, that we should support them so that we can use them. So that's the way to go on the future. Just a little bit in interim in particular. So they specifically went out to develop sulopenem for specific infections. And they got regulatory success, which cost them. But again, as I said, the high costs to get into the marketplace and our jobs not to use these new antibiotics basically led to failure. So clinically, they developed a great drug, but from the perspective of a commercial success and viability, nope, no way. So innovation is now being limited because we don't have people who want to look at it. And if you really look at our antibiotics that we use, we really don't have a lot of new novel antibiotic classes. We keep manipulating the ones that we have. And even in the new drugs in the pipeline, as I'll show you, these are basically a lot of the newer drugs are having beta-lactamase inhibitors, new ones, being attached to our older drugs. So we're working on our older drugs and only beta-lactamase inhibitors. They are making some advancements in those, but that's still classes that we know about. We're not identifying new ways to kill these bacteria, which would mean that potentially the resistance would not develop as quickly because it's a whole new mechanism of action. And did I turn it off? Oh, no. Okay. So as I said, we need to evolve beyond the beta-lactams, and we have to look at developing these new compounds that would be going after these very difficult agents. And so here I said agents that could reach the marketplace in 5 to 10 years. I made it up. I have no idea. I use 3 AI programs to actually give me which 10 antibiotics they thought would be developed within the next 10 years. I didn't get the same answer from any one of them. And actually, I was even afraid they made up something, which I did find. I would see something, it would say it would be in phase 3 development, and then I would go back and look myself the old way, and I would see, no, it's only in phase 1 development. So when they tell you that AI, if it doesn't know something, it will make it up to fill the gap, it does. But still, these are the things, the antibiotics that I will go over in the next few minutes are the ones that I think will make development. So new antibiotics, different mechanisms, looking at DNA, ribosomal inhibitors, DNA inhibitors, membrane disruptors seems to be a very big area that people are trying to focus on. And then there's the non-traditional approaches. Phage therapy, you probably all heard about that. It's a lot of talk about that. Development of monoclonal antibodies. And what I actually think between phage therapies, the microbiome therapies, not just for C. diff, but for looking at other infections like the urinary tract infections and so on. So a lot of neat things that are going on. So I want you to memorize everything on this slide. So this is for your reference to go back, but these are the different products I'm going to talk about. It gives you who made them, their target, and so on, and a likely time frame for approval. Like I told you, remember, this is my opinion on when they might be approved. So this is the framework I'm using to look at the rest of the presentation. So the first thing you do when you look at a new drug is I go on Google and say, how do I pronounce it? right? So this is solurabalin. And so it's a first-in-class macrocyclic peptide. So it's a peptide antibiotic. They're trying to look at it for HAP, VAP, and bloodstream infections. And it's actually, this is designed specifically for Acinetobacter resistant infections. So this is a pretty cool one. So it's developed solely for this infection, which, right, we really generally don't see it that much. It's more gram negative. A drug is developed for gram negatives and it has activity against everything. And it demonstrates very good activity against crab infections, especially against the polymyxin type resistant infections. It's a novel, so what we want, and it is, it's a novel mechanism of action. So it inhibits transport of lipopolysaccharides, and it targets the LPTB2 FG transport complex. And basically what happens is that it prevents the lipopolysaccharides from getting out of the bacteria, stays in the bacteria, and it leads to killing the bacteria. So what you'll see is either outer membrane disruption, and then the organism dies pretty quickly. So looks to be very good. Here's a pretty diagram. Basically, look at the bottom right. It just shows you that it's blocking. By the way, I took this out of an article in Nature, and I asked AI to enhance it a little bit to make it more colorful. And did a good job without screwing up anything. But the bottom line just shows you that you have the buildup of these lipopolysaccharides, which puncture the membrane, which leads to very rapid death. So what is the early data that we see? So the early clinical findings, good kinetics, good safety profile, very good microbiologic activity, just the way they had designed it, effective in animal models and in vitro, and is currently in phase 3 studies. So we'll see where that leads us to. So that's good. So, but the good news is it's in phase 3, which means that hopefully at some point they're going to be able to take it to the agency for an NDA pretty quickly. Now I want to switch to a different beta-lactam inhibitor. This is taniborbactam. It's a broad spectrum beta-lactamase inhibitor that we will see a lot about. It's basically a bicyclic boronate compound, and they're going to take taniborbactam and attach it to a lot of the presently available beta-lactams that are already out there. So we'll see a lot about it. This has a dual action. It's a reversible inhibitor of serine enzymes, and it's a competitive inhibitor of metallo beta enzymes. Whoops. And these are the different enzymes that it has activity for, and some that it may or may not be effective for. In this case, one of the further along is the combination of cefepime tenaribactam. And when combined together, we see activity now for CRE and for carbapenemase resistant Pseudomonas aeruginosa. This is just I thought I'd throw in a pretty graphic. I like the way it looked. It just kind of shows you where the different enzymes that this compound could have activity against when combined with another beta-lactam. So this compound had phase 3. Its registration study was called CERTAIN-1, in case you want to kind of look it up. This study was a study for uncomplicated UTIs or acute pyelonephritis. Okay, question for the group. Why is almost every new antibiotic, what's the disease state that they study and get approval for? See? Not CAP, UTI. Why? That's right. It's cheapest one to do. But none of them want to do UTI, right? They want to do HAP, VAP. So there's two things that's going on. One, they want to get your drug in the marketplace so that physicians can write for it off label. Number two, this is the beginnings. They'll get to see how well their drug is doing, and then they will start investing more money looking at the big ones, HAP, VAP, or bloodstream infection. But anyways, and that's my editorial comment as on the side. But anyway, so you can see, which is interesting, that the cefepime taniborbactam in this trial was considered to be like non-inferior or superior to meropenem. And if you really think about it, we don't really see superiority in many anti-infective trials. So when something like that happens, the first thing I ask myself, did they do the study the right way? Because it is so rare to see superiority in these small kind of clinical trials. But at least they got it from the FDA. Safety profile, nothing for us to be unexpected, basically with just beta-lactams. This was jointly developed by two smaller pharmaceutical companies. Malenta used to be its own company, then it got picked up by another company. By the way, that's what happens with all these small companies. They either go out of business or they merge with somebody or somebody else picks them up. And as I said, it was developed for UTIs, including pyelo, but really they're looking for broader spectrum diseases like VAP. So this drug was developed, went through phase 3 trials, and submitted to the FDA for approval, and the FDA didn't approve it. Right? We need new drugs. FDA didn't approve it. Not in this case because of efficacy, but in this case, they didn't approve it because they didn't like the manufacturing processes that the pharmaceutical company had. So you got to remember the FDA is not only looking at is the drug working in people, but how is it being handled by the companies in their manufacturing facilities. So this drug has been stopped until the company can come up with mechanisms to prove to the FDA that they can make this drug safely for the use in people. So it's almost made it, but quite didn't. Here's another inhibitor, nacubactam. This one is a bicyclooctane, and it's a beta-lactam inhibitor made by a Japanese company, uh, and it has dual mechanisms, inhibits class A's and class C's. And it also has activity on penicillin binding proteins type 2. So it's got multiple mechanisms to help with treating resistant infections. Just to give you an idea of what the target pathogens that the company is going to be looking at because of the activity of excuse me, of this inhibitor. So very broad, very wide. So could be very interesting. There's been some phase 1 studies. The kinetics looks good. Everything looks predictable. Seems to be well tolerated. Phase 2 studies also have been done. And the company is doing an interesting thing. It's doing a unique pathway towards development. They've combined the drug with cefepime and azathioprine separately. So you got a azathioprine nacubactam and a cefepime nacubactam, and they're developing them at the same time. You don't really see that too often, right? A company usually does one at a time. So that's pretty cool. And it's got very good activity and high antimicrobial eradication rates in their phase 2 studies. Safety seems to be pretty good. Nothing has popped out to be bad on this. And it successfully has gone through phase 3 trials, again, developing it for acute pyelo. And so there are 2 studies. I only highlighted one. This is the phase 3 integral 1 study. So you can see the safety. By the way, it was compared in these trials to imipenem cilastatin, where it was non-inferior. And again, this is statistically superior popped out on this one too. And so it looks like it's a pretty good compound. Infections that they're going to go after, as we would expect, as these target pathogens. So once it gets approval, we get to use it, the company will start going back for there. The drug, this one I don't quite understand. The FDA gave it fast track designation back in 2019, but nothing's happened with it in the USA yet. The Japanese company just submitted their NDA for this in Japan. So I got a feeling we're going to have to wait a couple of years till it goes through the Japanese process, and then the company. Don't forget this is a Japanese company. So then they'll come to the U.S. or maybe they'll partner. That's the other mechanism, trying to partner with a U.S. company. And so I'm going to assume we're going to see this in a few years being submitted to the FDA. Now, this compound is for staph, aphabicin. MRSA is obviously a problem that we all know about. Again, this is a novel antibiotic, so this is good. So we're not talking about reformulating an inhibitor or a beta-lactam, but this is a new class of compound. It inhibits FABL, and that's involved with fatty acid synthesis in bacteria. And basically, it kind of does the same thing as we talked about with the first compound, blocks the ability for the fatty acids to get out of the cell. The cell starts to die because it can't get rid of the fatty acids. And this is also very interesting because this is a very narrow therapeutic agent. Specifically, seems to have almost only activity against gram-positive MRSA, MSSA, and MRSA, and coagulase negative staph. So, and again, the reason I highlight the specificity of certain drugs is because we think that maybe that would be sparing from getting the side effects, like we know that most antibiotics cause C. diff infections. So if we get a very fine-tuned infection drug, so we can avoid that problem. This drug is available both orally and intravenously as they're doing their development. It's a prodrug, and it's in the body, it's converted to aflabicin desfosphono. It's in phase 3 clinical trials, so that's good. And they're comparing it, this phase 3 trial is comparing it to Vanc and linezolid. They demonstrated non-inferiority. And in this trial, about 50% of the people had MRSA at baseline, very, very good eradication rates with MSSA and MRSA, and excellent bioavailability. So it was a good IV to PO switch. So that would be a neat compound to add to our gram-positive armamentarium. Advantage, as I said, narrow spectrum, so maybe minimal effect on the microbiota in general, and potentially sparing us from other infections. The next part of the talk that I want to get on are some of the neat non-traditional therapies that are out there on the horizon. Bacteriophages, monoclonal antibodies, and antimicrobial peptides are compounds that companies are looking at to develop to have new drugs for us to treat these urgent or critical infections. So people know about bacteriophages, and one of the things that I really love about medicine and in science, when you think you have an idea, the first thing you need to do is go back to the literature to see, did anybody else have that idea first, right? Bacteriophages were being used in the early 1900s. And so now we're looking at bacteriophages all over again. It is undergoing a massive redevelopment worldwide because of the ability for their effectiveness that we could see then that might be able to help us with multiple MDR bacteria. Initially, they were looking at it for dysentery, but what happened is that I think this is one of those neat topics where people started looking at bacteriophages, but as soon as penicillin hit the market and everybody focused on developing new compounds like that, we in the West said, well, bacteriophages don't make sense. But whereas in Europe, especially in Eastern Europe and in Russia, they maintained doing bacteriophage research throughout this whole period of time. We in the U.S. and the Western world, we stopped and we only concentrated on either new antibiotics from natural sources or developing antibiotics synthetically. And so here's an interesting tidbit off the topic. Some of my past fellows will have heard me say this, but when we were in the early phases, like in the 50s and 60s, when people were trying to develop new antibiotics, where did we usually find these new drugs, these new compounds? Like where do cephalosporins come from? How do we identify them? By the way, I have a degree in history of science too. That's why I have to talk about that. Otherwise, my parents think they spent money on a degree that was useless. But well, they found it off a sewer plant in Italy. That's where they found the cephalosporin A compound. So when they realized that all these compounds could be are in nature, I'll never forget it. One of the times I was talking, this was back in the 70s or 80s, to one of the reps in Abbott. And we were talking about drug development, and he was telling me, when I first got hired by Abbott, they gave me a kit. And it said, when you drive across the country and if you see some goop on the side of the road, stop and pick it up. put it in this test tube and send it to the company. And Abbott, from that, they developed a library of 1,000 macrolides. So, right, we use macrolides for antibiotics, we use macrolides for transplant patients. So but they collected these things from all over the world. It's really a pretty interesting approach. So they were part of the drug development process. Sorry, I got sidetracked there. But so with the issue with bacteriophage therapy, although it looks pretty good, it's difficult to use. And by the way, in the United States, we cannot use phage therapy. There's only 2 centers in the U.S. that are actually working on this. If you think you want to use phage therapy, then you have to contact the FDA, and then they will put you in contact with these companies. What I don't really quite get yet is that I don't know how much you know about it, but you know how long it takes to develop a phage. So what you do is you develop a phage specifically against the organisms that's infecting you. So how long does that take? That could take 1 to 4 weeks. So, right, so think about it. Someone's is in the unit, they've got a multi-drug resistant, they're going to be dead by the time we create the phage to treat them. So, although they're out there in Europe, though, they have libraries of phages, or I call it a phage, it could be a phage. So what they'll do is they send the organism to the company in Europe, and they'll look through their library of phages to see does it match. And you got to be careful with phages too, because they can develop resistance. So what they'll do is they'll make a cocktail of phages. So you'll have, because the phage attaches to the organism, so it's a specific organism. So it's designed, it's like a virus. It attaches right to the organism, hijacks the machinery, it replicates, causes lysis, releases new phages. And it's, as I said, the idea of it is cool because it's developed specifically to attack the bacteria that is in the person causing the infection. And I said the issue is resistance because it can occur pretty quickly, but then that's why the cocktails are there. This is just a nice little graphic I threw in there to show you what it does. So it's precision therapy, personalized treatments, which are cool. And as I said, this is growing worldwide. I think they're going to be trying to figure out how to do this in a more rapid way, especially for our very critically ill patients. And they're looking at these types of infections specifically. Now, another cool area, I think, for future development is microbiome manipulation. And I think we're most familiar with microbiome for C. diff. And so the idea is changing the ecology within the person, or it could be topically as well, because it turns out different parts of our body have different microbiomes. So the idea is how do you manage those to get a positive effect? We're most, like I said, we're most knowledgeable about C. diff, but people are looking at decolonization strategies and prevention of getting this. And what I think is interesting, people are looking forward to using microbiome therapies to try to help people in the unit who are colonized with resistant bacteria. Interestingly, I wrote a paper back in the early 90s on trying to do something like this, except we took the opposite approach, which was wrong. We gave people antibiotics to kill everything in their gut to see if we could get rid of everything. Right, but they were populated anyway, so our approach was wrong. But it was but now we're looking at these colonization ones. The examples are live biotherapeutic or manipulated products or engineering products. I always have a joke, and maybe it's not appropriate, but I said the only time I'll ever take blank from my wife is if I get C. diff. So interesting topics that people are looking at are recurrent UTIs, and they're in phase 1 and phase 2 trials. So basically, the bacteria are either coming from the vagina or from our intestinal tract. And so the idea is, can we use them, correct the microbiome and populate it with mostly lactobacilli to try for recurrent UTIs to prevent them from happening. People are also looking at this and vaginal type infections, trying to repopulate within a more appropriate microbiome. And then we have looking at these transplantations to try to get rid of MDROs. This is going to be a very interesting field going forward. I'm going to be interested to see how it develops. There is a hiccup. I don't know if any of you play around. I shouldn't say play around with C. diff. Are interested in C. diff, but there was a company called Open Microbiome that existed there, where if you wanted to do fecal transplants, it was a consortium of universities. You go to them, but they were just closed down by the FDA. So the only places these are really being developed now are by pharmaceutical companies. So going forward, we're going to see, is there going to be enough interest with them in looking at these? I want to switch tack a little bit to antivirulence factors and antibiofilm agents, because these are going to be a major potential area of breakthrough for us. So with antivirulence, we're developing techniques to bind the toxins so that we're not causing problems. And one novel drug, which is NeoX-101, basically it aims it gets into the biofilm of the organism and allows the deconstruction of the biofilm so antibiotics can get to the organism, right? A lot of our resistant organisms have biofilms which protect them from our antibiotics. If we can get through that, then our current antibiotics can be effective. So, so that's going to be a very interesting look as well. Monoclonal antibodies, I don't know where these are going to go. We've had something like bezlotaximab, there's a couple out there. The idea is, we design a special monoclonal antibody against some of these MDROs? I don't know where it's going to go. I would think because of the difficulties we saw with our first monoclonals, might be more difficult, but if they develop something that's really effective, that may change things. And again, the monoclonal is only designed against one organism, where many of our antibiotics have a broader spectrum for gram negatives. Now, when I was looking at this, and I forgot about this, another thing is looking at antibiotic peptides that basically punch holes through the cell wall. And so I thought that's a cool idea. And then I forgot, we already have drugs that do that, that we can model this after. And so, like polymyxins or daptomycin, these are antimicrobial peptides. You punch a hole in the bacteria, and the bacteria is obviously going to die, and it's actually rapidly bactericidal. And then there's some other peptides that we know about or that we're looking at. There's the peptides called defensins, and these are on our skin. So people are going to try to look at these to see if we can do something with them. Cathelicidins, it's a prominent one. This is in us again. And these peptides will bind to some cells. They're stored in cells and then released when infection occurs. I've never really heard about this one, hacetidins. And they're potent antifungal peptides, and then bactericidins, which we've all heard of, which can kill different bacteria. So looking at these peptides that can be cidal is going to be another way, and maybe they'll be in combination approaches. We're not going to. But the cool thing about these, as I mentioned earlier, is they work on biofilms. They're very good on biofilms. There's a lot of research showing that these newer synthetic peptides can break through the biofilm of these bacteria, especially MDRs, and allow us to use standard therapies that to treat these infections. So I very quickly went through just a small number of drugs that I thought, but it's not just needing the new antibiotics. We have to understand what's going around them from a drug development process. We have high research development failures. Only about 30% of these compounds might make it to a clinical trial. This was an interesting statistic. Worldwide, they estimate there's only 3,000 researchers who are working on anti-infectives. That is 10 times fewer than the number of researchers around the world working on oncology compounds, right? So, and again, it gets back to, I hate to say it, where do you make more money? An antibiotic or some of these other agents? And here's another interesting statistic. Over the last 10 years, industry has spent $26 billion trying to develop new oncology products. Industry has only spent 1.5 billion roughly looking at new anti-infectives. So you can see the way we develop our marketplace is geared away from this real big problem that we have, right? That is killing more and more people every year and is getting worse versus the companies looking for the different things. And as I said earlier, what do you want? Another Z-Pack drug, or you want another Viagra? Where are you going to go? Right? Everybody's every commercial I see now, they want everybody on Viagra, men, women, you name it. You just take it. Uh, my 7-year-old granddaughter knows the names of drugs, right? Because every commercial that comes on is some other new drug. So it's like maybe she'll be a pharmacist. I don't know. And so we have to figure out ways so that companies are going to be profitable if they're in the marketplace. What's another challenge? We are, right? What do? We have stewardship programs. And we right, in our hospital, basically, when a new drug comes out, we don't even really add it to the formulary right away unless it's some breakthrough drug. We just wait. And other places you'll put it on formulary, but it'll be restricted, right? You got to go through 10 layers of locks before you're going to open up to use a new drug. So it just sits there. And we're going to use, an older compound. So that means the company's not making any money. And as I showed you with those earlier companies, they developed the drug, they got to the marketplace, but they couldn't recoup the amount of money they dumped into it because they want to go to higher areas. So bankruptcy of companies. So if you're a company looking to develop a drug and you see that drug companies before you failed, you're not going to go into the marketplace. So in Europe, they've been approaching this, realizing that scientific success does not equal commercial success. So they're trying to figure out how to support companies develop new compounds for us. So I'm hoping that we're going to see some of these compounds that I talked about in the next 5 to 10 years. Some of them are very exciting, and if they get developed and they get out there, we also need to have a way where our government here in the U.S. is being supportive of these companies that are developing it, so that we will be able to use them. And with that, I want to say thank you, and you were very attentive. I would say if there's any questions, you can ask. I don't know if I have answers.
Could you use maybe a orphan drug approach, like so that you do support it from some either NGO or government source to say, look, we're going to be doing a great thing for a very small amount of people. Let's be seeking support that way.
Yes, that's in case you didn't hear her question and comment. Is there a way to incentivize and maybe use the orphan drug approach? One of the things I've been lucky in my career, I was involved with the development, as Shannon has talked about, a drug called paromomycin. It's an old amino glycoside developed in the 1950s. And when gentamicin came out, everybody said, "Oh, gentamicin is safer than paromomycin," which was wrong. But the paromomycin went on the shelf. So jump to the probably this is about in the late 90s. One of my fellows was a pain in my ass. Who came in and he said he wanted to do some research and it was on this drug. And I said, go to a lab and start working on it. And he came back and he starts telling me how the drug is effective against every gram-negative organism that we can see. And so what did it lead to? One of the other things it was active against was visceral leishmaniasis. So then as we're developing it, I teamed up with a small generic company and they were happy because they were going to get a drug to add to their portfolio. And we figured out the drug was going to cost $1 a day to treat people with VL, visceral leishmaniasis. And then all of a sudden, some of the other people in our group goes, "Do you know that some people in India don't make $1 a month? So who's going to pay for this drug?" So it was like, oh, we got stymied. So what happened is I partnered with the WHO, and then they went to, there are nonprofit pharmaceutical companies. So they the university signed over the rights to the WHO, and the two entities developed the drug, and now it's available on the market and is being subsidized by different companies. So that your approach is the right way to do it. We did orphan drug. That's how we got some initial money on it to get to a point. So we're going to have initially these kinds of mechanisms to develop these new drugs, and it can work, but it took 10 years. It took a long time for us to get that product out. I was lucky. Other questions? All right. I hope traffic's not too bad getting home. Thank you very much. I appreciate it.
But yeah, it made me laugh because I was like, oh, that's just a second. It's like one or two years old. It was, no, he was sick. He was sick. Yeah.
I'd say that was excellent.
Oh.
You didn't want to do it again? Sure, that's such a good job.
But I said, every time I was moved, I was, right, I had to take the drug out of the presentation.
But it worked out. You got it.
They're really hard to one and stop both.
Thank you.
Yeah, well, no, he's got to do
And what are you doing now? You're
I'm at Pfizer now in the COVID management space, so I've passed a little bit. So past like 4 years since the pandemic. But I mean, I think they might potentially like do another reorg and chop.
Are you gonna start on that?
Especially when Danziger doesn't do so well, it's in phase 3 now, and but I'm thinking like in the summer with, not that I'm rooting for COVID, but I don't, they need to enroll 2,300 in a new trial, and like they're not. So I'm just like thinking of job security, and I was like, that's
So
very.
I don't worry, in the US, the way the HHS is going, we
Yeah.
will have a great COVID outbreak this winter, I guarantee it.
Okay.
Right, what is it? Kennedy's not interested in doing that.
Yeah. So if you were me looking for a position, and I came from a company before Pfizer, after Rush in Chicago Department of Public Health, I had like seen a STARS position,
Oh, yeah.
and then ID by DNA got acquired by Illumina, which is like a precision metagenomic.
So, you know,
IDS
that's what I
company.
thought, but everybody's buying everybody.
Yeah, and then I had an offer from Insmed and Pfizer, and I was like, Insmed's another on fire startup company, Pfizer is a big pharma company, I don't think I'll get another chance at them, so I picked the Pfizer job, and then this week 2 at Pfizer, there was a reorg. You never know.
If you can stand all the reorgs, obviously being a bigger company.
Yeah, I've survived so far, we've had enough cuts so far, so, but if I were to write a prompt in AI to try to predict which pharma company would actually make it, how would you word it?
Who's got the better drug pipeline?
Yeah.
Right, like that's how I would try to say it. But then, like I said, you gotta check I would check as many of the AI databases as you can, because they're all different.
That they're
Yeah.
coming out and then they're.
Well, funny.
So you think.
Good to see you, thank you so much.
Oh, thank you, Randy.
But they're going to phase 3 now, but like. Oh, she hasn't started. I mean, overall, it looks. She's got, yeah, those are all.
You actually did show up.
Yeah, of course it. I was just saying, so there for. So Sarah Baldwin also actually. Pretty good there. Yeah. They presented. Yeah. You guys see Rock now, Mari and the other kids.
Yes.
Okay. They'll be, if they are successful with phase 3, I think they'll be.
Well, I do. We get probably, they're also stuck, the agency, like something that was.
Maybe a little bit faster for them too.
Yeah, I didn't read the letter,
You know, I don't mind
Okay.
taking Western Rockford because it's a known
They
entity.
can figure it out.
You know,
Tanny were back and they much.
as much as I like to say.
Right, they have a drug, looked like it was gonna be good, but they just decided.
And I said, well, look at,
Right.
So they were like, no.
No, yeah, why fuss around, but we'll put our money somewhere else. But see, there's another, one. You also gotta worry that if the FDA has a deal for you.
Yeah.
They won't let go.
Yeah.
That was, you know, the company, like that small generic company I worked with.
We had a lot
So they got into a hissing match with the
Yeah.
FDA about manufacturing issues. And so after that, the FDA, no matter what company did, the FDA said, no, they do this, and they make it.
He got one.
So
But
that
he
was
was,
like, opened my eyes to like,
They
they wanted to, they wanted to jack you around, they'll jack you around.
Who knows what it's like now? There's so much change, so there,
Right.
it's hard to know what to do.
Yeah, so, yeah, no, we gotta wait
And
until
then we put him on.
some other president comes in and. So we gotta.
Yeah, I saw one of
Oh
them.
my gosh,
And I was like, I wonder if someone's looking for it.
that was me.
You licked the
Yeah,
cap up.
I
And I
was
said,
I'm save the boundary waters that are currently on fire
Yeah, you're
in
out.
Minnesota, you
Yeah,
know,
close,
yeah,
You know, that's,
yeah, they
I
evacuated.
don't think it's only the third time.
It's crazy. I don't, anyway. Yeah, this was about drilling for natural resources, but it's currently on fire, so relevant.
Yeah, no, I'm official.
Oh, I grew up in northern Minnesota, so.
I was in phenomenal, just.
Oh.
Where's your?
Duluth,
Oh.
yeah, my
I
parents.
always found this.
Oh, really? Are they Bob Dylan too? I don't know.
Of course.
Northern Minnesota.
They all moved down to Chicago because nobody went there.
Oh, it's fun to visit. Though. So pretty. It's good that my parents live there, so I get some nature. It's been less of Yeah.
That's still nature. It is,
Yeah.
it's actually on the website. Tomorrow morning. I don't
The
know.
whole state's like covered.
I know.
Oh,
It's like that. Yeah. I looked on my window and I was like,
Okay, three blocks and I'm very cool.
I'm from Northwestern. Oh, I'm in Northwestern, downtown.
So, yeah. So,
Yes. Yeah.
yeah, I was. I haven't seen it.
You see the master.
Look up,
Not too much sunny fire.
Chris, they trained development.
No, sorry, don't you just.
I know.
That was. Honestly, impressed me get it now. I know, I don't know. Oh, it's cold.